Yeah, hi to all participants, and thanks already for joining us today to our webinar about transforming tissue research with spatial omics workflows. I'm sorry. My name is Frank Krebs. I'm responsible at Avantor for the furniture and lab equipment business in Europe, and a warm welcome from my end, and also a warm welcome from our colleagues from Leica, which are putting you through that presentation and that learnings today. But before we start, just want to share some housekeeping notes. So we appreciate any questions you might have, and that you can, or they can you raise on the chat area, especially they are not in the chat area, but in the question area. You see that happening, that little icon with the question mark within on the top right of your screen. So if you have any questions during the webinar, just raise them. We take care about them. Either on the end or if that is aligned to a slide from Daniel or either Michael, I will raise them in between, but don't hesitate, as said before. So you don't have to write anything down during the webinar because you get a full recording, including all materials shared in this webinar right after the webinar, together with a small survey where you can give us some feedback about what went well, what should be improved, what you want to see next time from Avantor. But also Daniel has prepared some polls during the webinar, and one poll he will raise verbally and asking you for giving us some feedback within that small survey we send out right after the webinar. If you need any certificate of attendees, so please send us that wish as well. We are happy to fulfill that wish for this certificate of participation in that webinar. Having said that, I will hand over quickly to Daniel. Thanks already, Daniel, and the stage is yours.
Thank you very much, Frank, and a warm welcome also from my side. Thank you, everybody, for taking the time to be with us for this hour where we would like to present to you how we as Leica can help you in your omics workflows and how microscopy can help you transform your research. So let me start. All right. So first of all, spatial biology, or what does that actually mean? And why is it important? Well, if you are looking at tissues or cells, those are complex structures that have a lot of subcellular components that are interacting in space and time with each other. And I always like to compare it with Legos. My kids and myself, we are huge Lego fans. And just imagine you have a box of Lego pieces without a manual. That would make it very difficult to get to the full figure that you would like to have in the end. And it's kind of similar with spatial biology, where we are putting the many different proteins and other components in cells in context to the whole tissue or organ in the end. And this helps you then, of course, to go beyond, let's say, a small snapshot that you can normally get with other methods like transcriptomics, proteomics, mass spectrometry, and others, which give you a lot of information, but which is either averaged out, let's say, over whole tissue, or which is just one point in time. And in microscopy, we can put all this now into context of the whole tissue. We can provide the manual, so to say, for your Lego, which then helps you to create the bigger picture. And since we're talking about microscopes today, at Leica we have a huge portfolio of different microscope systems, and today we want to focus on a few of them, which can also work together in a workflow, especially in spatial biology. And you can see them on this slide here. Maybe it's now a good time to raise our first poll, which you can find somewhere on your screen coming up now.
Yeah, it's coming up in a second. So I will launch the poll
it's
right
coming
now.
In a second. Yes.
So.
Yes. So a good starting question would be, are you currently using microscopy in your workflow? Yes or no? Let's see how many people are already working with microscopes. let's give it a few more seconds. Okay, 50-50 at the moment. Not that many participants so far, unfortunately. But okay, some of you are working with microscopes already, some are not. So let's hope we can change that a bit in the future after this presentation. And to be a bit more specific now, so what do we want to talk about in this presentation? I will start with giving you some examples of how our systems and products can help you with preparing your sample for your spatial biology workflow. So for instance, for cell culture check or checking your tissue section. Then once you have prepared your sample, you might start with a subset of dyes or proteins that you want to look at in one sample, which you can do very well on our Mica microscope, which is the second one that you see here on the screen. And after that, you might go with your full-blown up to 60 different marker hyperplex imaging, which you can do on our so-called cell dive microscope, which my colleague Michael will tell you more about. And of course, in the end, you need to extract also your analysis, your insights from the samples that you have prepared and from your imaging. And we can then also tell you a little bit about this part at the very end, which is also what Michael will talk about. So let's get started with the first step, checking your cells or your tissues before you actually do your staining or start your acquisition. And this typically involves a couple of steps. So first you want to seed your cells or prepare your tissue and just have a brief look if it's looking well, if you have enough cells, if your tissue section is the one that you want to continue. We do have some workflows embedded that help you doing that and also do some automatic measurements, for instance, cell confluency checks. You can then, of course, document those data sets and then continue with your scientific experiment. There is a small video here on the slide, which I hope also comes with sound, which shows you what kind of system we have
Turn on the microscope.
to do that.
Place your cells on the stage. Choose the desired magnification by rotating the objective nosepiece. Use the focus knob to obtain a sharp image of your sample. Find an area of interest. Take an image by clicking the camera icon. The images are stored in the gallery. Go to the gallery, find a stored image, and double-click on it. Turn on the microscope.
Okay, so the microscope that you just saw also has a name. It's called Matteo, and it allows you to do exactly what you just saw. So check your cells before you continue with your staining or your tissue section. You can do that in bright field or phase contrast, as you see here in this image. And it also comes with some very neat and easy to use workflows. You just click on confluency check and it will automatically tell you how many cells you have in your dish. Of course, this part of the workflow also comes with some typical pain points that users have, which you can see now here listed. And thanks to Matteo, we have addressed these most common things. So it comes with a very intuitive and user-friendly software interface that really everybody will be able to use. Also, more complex contrasting methods like the phase contrast that you have just seen can be used by everybody because it comes without the need for manual adjustments, also without the need to adjust other things like light source and camera, because this is all automatically handled by the microscope. And this makes it then very reproducible as well. You can just reload your imaging conditions so that once you have established a workflow in your lab, everybody will be able to carry them out. What if I also want to have fluorescence directly? There is now, since this year, a big brother of the Matteo that you have just seen, which is called Matteo FL, which is able to do exactly that. So here's also a picture how it looks like. You can either put it on your bench or under the hood. It's very small and compact. It would fit under here as well. And in addition to the Matteo with only transmitted light, it also comes with fluorescence. 3 different channels actually, blue, green, red, which can be very easily and automatically set up. It also comes with a few more analysis options, again, very easy to use. You just click on them. If you, for example, also again want to measure confluency or count how many cells you have there, or since it also comes with fluorescence, there's also a tool that allows you to quantify your cell transfection efficiency. So that would have been maybe the first start in your spatial biology journey. And from there on, you have then prepared a sample already, and you can take it to the next microscope and next system in our workflow, which comes in the following slides. But before I start with the details, maybe it's again a good time to raise another poll question, which is...
For sure, Daniel.
Yes.
Just give me a second. I will launch them right now.
I'll wait a second.
Yep.
So what are your biggest pain points in your tissue imaging experiments? Or what would be the most important points for you to solve for you in this workflow? Is it all about the resolution that you can achieve? Is it that you struggle with the image analysis routines that you have? Or is the image quality that you are generating not good enough? Is it about the acquisition speed? Or is the number of labels that you can have in one sample the limiting factor? So far, not too many answers. Well, okay, 2 coming in. Resolution,
I think
all of them.
I think it's worth to mention that the participants can scroll down, please, for more options, for more answers. You can scroll down through this drop-down menu through the different answer possibilities. Maybe that's...
All right, more coming in. Let's give it
quality and resolution so far.
Image quality and resolution seem to be topics that the people that have answered here are concerned about. So let's see what we can do about that. And with this, I would like to introduce you the second system that we have put together here in this presentation and that you can use in a workflow for your spatial biology experiments. And the name of this system is MICA. It looks like as you see here on the screen. And we actually call it a micro hub because it connects many different imaging modalities in one system. It's also an incubator, so also suitable for samples that need higher temperature or CO2 incubation. And what we focused on when implementing MICA was that it's accessible for really everybody, so easy to use, that there are no constraints between different imaging modalities. You have everything in one system, and that it comes with radically simplified workflows that you can simply call up and use from the acquisition to the analysis. So let's have a bit closer look into these 3 different topics and what they mean exactly. So access for all, because we try to eliminate over 85% of the steps that are typically required when setting up your acquisition on a high-end fluorescence microscope. And here's an example about that. So again, fitting into the spatial biology context, you see a tissue section labeled with, in this case, 4 different fluorophores. And typically, if you would have to set this up manually, even as an expert user, this would take you a lot of steps to get to this image. And now with Mica, it's 85% fewer steps. And the same is also for the amount of time that it takes to actually acquire it, which is around 1/3 less than if you would do that manually. And the hours that it takes to train new users, and really everybody in the lab, is also reduced by half. And this is thanks to different intelligent tools that we have implemented in the system. So it's extremely user-friendly and actually takes care about a lot of settings by itself, which then includes also the imaging itself and the setup of your experiment. The second topic is that there's no constraints. So you have really everything that you need in one very easy to use system. And you can also get up to 4 times more data, meaning multicolor data, which are 100% correlated. And what does that mean? And why is this important? So Mica is on one hand a wide field microscope. And normally on a wide field microscope, if you want to work with multiple colors, that involves switching to different filter sets. And that, first of all, slows down your acquisition speed. And it also leads to the fact that if you have moving samples, you will never have 100% correlated data, because there's always this moving time in between, and you will always have a shift between your different channels. We have solved that completely in Mica. So you always have 100% correlated data. You can actually acquire in wide field acquisition mode up to 4 channels simultaneously without any time difference. And this really gives you then an uncorrelated spatial temporal matching of your channels. But that's not all. So there's even less constraints, I would say, because Mica is not just a wide field microscope. As I mentioned, it's also an incubator. It's a wide field microscope, which can also come with so-called Thunder method, which is our Leica owned and unique method, which removes all out of focus blur in your wide field images. So we call that method computational clearing, and it's completely integrated into Mica. However, it doesn't stop there. It can also be used as a point scanning confocal. So that's an optional thing on Micas, but you can have Mica as a so-called wide focal. So not just with camera-based detection, but also really with point scanning, laser excitation, and confocal detection. And all of that in one instrument, which, for instance, in your spatial biology workflow would mean that you could have, for instance, 4 slides with tissue sections with multiple fluorophores on them. Mica would allow you to actually automatically find them where they are on the slides, focus on them, and do, for example, as a first step, a very fast wide field acquisition to really image the complete slides, the complete sections in the whole sample area automatically. From there on, you might then say, okay, now I know where my sample is, I know where the interesting areas are, but my resolution is not high enough, which is also a topic that most of you have now just raised in this poll. So what can you do when that happens? So here's a small recording out of the Mica software, which shows you what you can then do at this point. So once you have located your areas of interest, you can, as a first step, switch from the 10x to the 20x objective. It's still 100% correlated, as you can see. You're still in the very same spot, but already with better resolution. From there, you can switch to an immersion objective, 63 water immersion. The water will be automatically added between your sample and the objective by Mica. And that's not all. From there on, you can switch from wide field to confocal acquisition mode. As you see, just one click, and you have not a wide field image, but a confocal image. And if you have now in this recording noted one thing, there at no point it was necessary to actually set up the acquisition parameters. There is a so-called one button, which then automatically adjusts things like detector gain, laser power, pixel format, acquisition speed, all these things that you normally have to set manually are taken over in an intelligent way by Mika. And just by choosing that you want to go from a wide field overview image to a high resolution confocal image, the system is actually doing that for you with 100% correlation, as you can see. Once you did that, you have data with much better resolution, better image quality also. And this is then something that you can use directly in an analysis workflow, for example, for 3D imaging. You acquire a Z-stack, you have multiple channels, and you can directly use the analysis tools that are built into Mika, also using deep learning, machine learning tools to get to your insights. And all this in one easy-to-use workflow. So with this, I'm at the end of my presentation almost. I would just like to mention that there are more microscopes and more systems that we have in our portfolio which can actually address your spatial biology needs. And just to show here on this one slide, we have a high-end confocal microscope called Stellaris, which, as we speak, since this month, also comes with a spatial biology workflow completely embedded in the system. And I think I have to reshare. Sorry about that. Which comes with a spatial biology workflow in the system, which is called Spectroplex, which we have just launched now at SFN in Chicago just this week. And here in this example, you can see 8 different biomarkers plus DAPI in one tissue section, imaged in 3D with the confocal at point scanning resolution, so highest possible resolution and image quality. And 8 is actually not the maximum, so we have already shown that we can do that with 15 different markers in one sample, in one go, in one imaging round with the Stellaris. Another system which is very useful for spatial biology workflows is our LMD, laser microdissection, which allows you to specifically cut out parts of your tissue for downstream analyses. And there was 2 years ago a very widely cited publication which used a whole workflow starting from a tissue section taken from patients, imaging this with a wide field microscope, for example, Mika, running deep visual proteomics, so image analysis on those images, and then deciding which are the interesting cells that we want to further analyze, which are the cancerous cells in this tissue, taking then the sample to our LMD, which is then able to precisely, with a laser, cut out the cells that you want to analyze and collect them just by gravity. And then you can take these cells and really on a single cell level do, for example, mass spec on those cells to get a protein profile. And this then really allows you to connect our spatial biology workflow that I've shown you some examples now here, for example, with mass spec analysis, which is something that will be very useful for things like personalized medicine, for instance, in the future. And sorry for the small confusion. Now I'm at the very end of my presentation, and I'm handing over to you, Michael. Thank you very much.
Thank you, Daniel. Just let me pull up my half of the presentation and I'll get right going. So hi there, everyone. Thanks for having me, and thank you, Daniel, for introducing the rest of the portfolio. I'm here today to talk to you about CellDive. CellDive is an answer to the question I have listed on the right side of the screen here, which is how do we approach experiments where visualizing many markers is critical to mapping the spatial landscape of the tissue? And I'd like to walk you through the background of why someone would ask this question and why you would need more markers than a traditional, say single digit microscopy experiment where you're looking at maybe 4 to 6 markers, potentially even more than what Spectroplex can achieve at 15. How would you visualize cells if you need to look at many different cell types using 10, 20, 30, 40 or more biomarkers. And cell dive is the answer to that question. But how did we get here historically, where people would be asking that kind of question? Cell dive is an attempt to thread the needle between two kind of divergent technologies, one of which would be omics technologies, and the other would be imaging technologies. And the benefit to an omics style technology like RNA-seq is that you get very complete information about the expression information of different genes in populations of cells. You can learn every RNA that's being expressed in a population of cells. And another method to look at cell identity is flow cytometry, where you can break apart tissue and learn the identity of nearly every cell in that tissue. The problem with these two methods is that while you get nearly complete information about the identity of the cells and their expression state in the tissue, because of disaggregation and lysing of cells, you lose their position, their spatial information, the context that they're in. Imaging approaches, microscopy, on the other hand, you keep all that spatial information, but there are often technical limitations on how many markers you can look at. So the identity of every protein that's being expressed becomes harder to access. And why is it important to look at the identity of every cell in certain tissues? I have here a little diagram of a tumor immune microenvironment. This is just to show you that as cancer research, for example, has progressed, we've learned that modeling cells in a dish doesn't really present the full picture of how cancer arises and progresses in response to treatment. There are many different tissues and systems that are intersecting at the tumor microenvironment, such as the tumor cells themselves, the immune cells that surround them, the extracellular matrix proteins, the vasculatures. So really, to understand and to treat complicated diseases like cancer, we need to know what the different parts are in the system. We need to know where they are and how they're interacting. And so we need to collect that spatial information as well as the identity information. So how do we do that? And what have been the roadblocks to achieving this? And here are just a few examples of things that kind of get in the way of collecting all that information. Traditional microscopy approaches, the color and marker selection can be inherently limited. If you're doing staining with antibodies, validating those antibodies can take a really long time, and sourcing them can be difficult and expensive. When working with tissue samples, such as cancer sections, those samples can be really precious, and a lot of imaging techniques are not preserving. So you can image those, and then you may have to throw them away. Proper imaging and doing these correctly often requires a lot of expertise, which not every researcher has the time or interest to develop. And processing and image corrections can be really time consuming to get good quality data that's ready for analysis. And speaking of analysis, data management for these large data sets is really hard. They take up a lot of space, and moving it around and working with it is a challenge. And actually analyzing, tens of thousands of cells in a single section of tissue is, I think, pretty obviously hard to do. So like I said before, a key insight in cancer research of the past decade is just how important this tumor microenvironment is. Because if you can understand all of the cells and systems that are in the tumor microenvironment, you can accurately predict patient outcomes, improve stratification of patient populations in your retrospective or clinical studies, and you can add to the understanding of patient molecular and genomic data. So, with all that I've just discussed, that kind of intersection of the omics approach and the traditional tissue imaging approach, we have cell dive. So to kind of walk you through the logic of how cell dive came about, imagine you start with the traditional tissue imaging approach, where you're working with FFP tissue blocks and you're sectioning those out and you're doing a chromogenic stain. You can really only look at 1 to 5 biomarkers. And if you want to look at more biomarkers, then you can use serial sections, but there's a loss in resolution there. So you get some spatial information, but the amount of proteins or analytes you can look at is fairly low. On the flip side, we have the genomic approach where you could just grind that piece of tissue up and look at every RNA in there, but again, the spatial information is lost. So cell dive, again, tries to thread that needle by taking those tissue sections and then staining them with an iterative staining approach that allows many antibodies to be built up on the tissue and visualized. And to simplify the overall workflow, we have a purpose-built imager with purpose-built software and inbuilt calibration to make the imaging process as automated as possible. The end result of this is the ability to image many biomarkers from a single tissue section. So this is CellDive right here on the table with these two researchers. It is a completely customizable, precise multiplexing tool. It can spatially map 60 plus biomarkers from a single tissue section at a single cell level. It's customizable. You can configure the experiments exactly how you want. It's precise. You can collect all the data that's valuable to you and trust your results. And it's proven. It's a combination of years of experience and 3 tested and refined technologies that are merged into one solution. And this is the overall workflow here. We start with FFPE tissue sections or tumor microarrays, and we prepare those according to standard immunohistochemistry processes. We bring those into the CellDive imager, and we select the regions that we're interested in imaging. And then we begin an iterative staining process to build up the biomarker images. And we achieve that by staining that tissue with 4 antibodies directed against biomarkers of interest, each of which is bound to a different dye. We then image those biomarkers, and then we inactivate those dye molecules using a gentle dye inactivation solution. We then perform some autofluorescence imaging to subtract out background, and then we repeat this process 4 biomarkers at a time until all the biomarkers in the study have been imaged. Once we're finished with that, we can perform image processing using analysis tools. We can use image processing that's built into the CellDive process and then perform our analysis using analysis tools. And just to give you an example of what types of information the CellDive system can deliver, here we have a colon adenocarcinoma section, and I'm going to show you a few different panels highlighting different biological pathways. So just to start, we have a mini panel of immune markers such as CD3 for T cells, CD4 for CD4 positive T cells, and CD20 and CD45 for other lymphocyte markers as well as CD8. We can then change gears and look at metabolic markers. So we have GLUT1, beta-catenin, HK2, HTR2B, and then change gears again. And now we're looking at apoptotic markers like BAX, caspase-9, BCL2, etc. So we're able to look at 3 different biological pathways all in the same tissue, all at the same time. And that can open up a lot of really interesting areas. Typically in a lot of biological research, we may be looking at just 1 or 2 phenomena at a time in a given experiment. With multiplexing with CellDive, you can look at 3, 4, 5 different biological pathways all in the same context and see how they respond to each other. So it really opens up tremendous avenues of research and analysis. These are all the parts that make CellDive work. I'm going to go through each of these in turn, but CellDive is a combined effort from validated antibodies, the process itself, the software, the imager, and downstream of the images, we have image analysis with AVIA. So I'll start with the validated antibodies. As you probably know, antibodies are a major point of failure in these types of immunohistochemistry experiments. They're a major culprit in the reproducibility crisis. And validating antibodies for any sort of staining protocol is time consuming and can be cost intensive. So to make this process as short as possible, with CellDive, we include a list of already validated antibodies, more than 350, that are ready to be used with the process to get biomarker panels built up quickly and experiments rolling as fast as possible. And when it comes to building out primary conjugates to use with CellDive, you have different options, and we are agnostic to how these work. You can pull antibodies from our list. You can validate your own antibodies using protocols that we will provide, and you can also use third-party kits as well to pull in these antibodies. So you have a gradient here between off-the-shelf and completely custom, and you can combine these as fits your needs. Next, I'll talk about the CellDive process. So the CellDive process uses a patented dye inactivation solution. It's been patented since 2009, and it turns off dye molecules in the sample, and it doesn't damage the underlying sample, allowing you to build up additional staining cycles with new biomarkers. It's validated for use with FFPE tissue sections and tissue microarrays, and it's compatible with cyanine-based dyes, which concludes the side eyes, some alexifluor dyes, so on and so forth. This is the overall workflow, kind of from beginning to end. It begins with study design, so acquiring your tissue, planning your biomarker panels, doing the antibody preparation. Then we have a slide preparation process, which will guide you through. This is standard IHC protocols. Then in the CellDive software, you can build up your imaging protocols. You can add slides to those protocols and create imaging batches. You can do your region selection using either a 2x or a 10x lens that are built into the system. And then we can go into those staining rounds that I discussed earlier. We begin with an autofluorescence image. We label with our biomarkers. We visualize those biomarkers, and then we dye and activate those biomarkers before repeating the cycle. I'll talk about the CellDive software next. The CellDive software has three main parts: study management tools, the image correction softwares, and the ability to precisely register images between rounds. So I'm just showing here the study management software. That's a major part of the process. It offers the ability to build comprehensive protocols that contain all the details of the multiplex study and guide the user through the workflow. And it works in conjunction with an integrated barcode reader that's part of the CellDive. Each slide will be barcoded, so you can keep track of multiple samples and multiple studies, multiple users, totally effortlessly. It takes a lot of the heavy lifting mental work away with the help of the barcode reader. These are some of the calibrations and image corrections that the CellDive will perform automatically using its software systems, such as objective centration, blank glass subtraction, flat field correction, intensity calibrations, distortion correction, stage alignment, image alignment, and registration, as well as autofluorescence removal. The end result is that when you've performed your biomarker imaging for all your biomarkers, you have fully corrected final stitched images that require no further user intervention to process. And the end result of all these techniques is the ability to achieve subcellular resolution of markers. For example, we'll show here Ki67 staining, and that's a marker of proliferative cells. And you can see here that we're actually able to resolve individual subnuclear foci. So if your study demanded it, you could quantify those foci or even measure their morphology. So that's the power of these image corrections. And if you're working with pathology labs, you may have run into a situation where they really want to look at a virtual H&E image, and that can pose a challenge for fluorescence-based tissue imaging methods because it's difficult to combine the two. And to solve that issue, CellDive also features a patented algorithm that can create a virtual H&E image from fluorescence signal, and you can provide these images to your colleagues and work together with pathologists to define ROIs and other regions that are interesting in the tissue. The CellDive has a very large imaging area. It can do whole slide imaging, 20 by 45 millimeter if you're working with the cover slip samples, and a little bit smaller if we're working with a click well, more on that later. And you can break the tissue up into regions of any size, shape, or quantity in software. So it's a totally flexible system that lets you capture as much data as possible. Talk about the imager itself. The imager features an integrated barcode reader for slide tracking. It's a 5-channel imager to accelerate your multiplexing. It's compatible with robotic arms and other robotics for walk-away imaging. And it features the patented Clickwell slide holder, which eliminates repeated cover slipping. It's engineered for precision, speed, and sensitivity, and it's based on a proven high-content analysis automated microscope platform. Like I said before, it's a 5-channel imager. It has a very large imaging area. It works through slide imaging and features the Clickwell slide holder to eliminate any cover slipping in the multiplex process. And the barcode reader automatically tracks slides within and between studies. In addition to its ability to integrate robotics, it also features robust hardware and software autofocus for reliable imaging. And this is the Clickwell. This is our patented slide holder to make these staining processes as simple as possible. In the past, for multiplexing studies, the user may have had to put cover slips on and take cover slips off to introduce staining solutions to the tissue. The Clickwell is a slide holder that uses an insertable insert to create a reservoir where liquids can be flowed in. It features reliable and repeatable slide placement that allows you to choose the staining option that's best for your research. You can stain manually directly in the click well. You can remove slides from the click well and stain them on the bench and then return them to the click well. You can keep them in there the whole study and automate staining using an automated pipetting system. You can remove slides from the click well and automate the staining using an auto stainer. And these are also compatible with robotic arms for automatic loading into the cell dive imager because they follow a 96 well format. And you can use multiple click wells to increase the scale of your study. And I'll just play the video one more time so you can appreciate how this works. The user here inserts the slide into the click well base and tightens it into place using these adjustable cams. Then takes the click well insert and clicks it into place, and that will form the reservoir, allowing them to add staining and wash solutions. Automation. Automating the staining process can greatly reduce the touch points of a multiplexing study and allow the user to look at more slides and get the study done faster. And by using ClickWell, it actually opens the door to a wide range of different automation choices, and you can choose the tools that are going to work for you in your laboratory's multiplexing approach. This is just to highlight, if you were to do the cell dive workflow completely manually, this is the touch points that you would have to go through. Each time you see an image of a researcher, these are the individual touch points you'd have. But we have partnered with a company called Advanced Solutions to produce this bioassembly bot 200, which can automate the entire staining process. It will automate entire cell dive imaging rounds, and that reduces this process to just one touch point per round. And I have a video here showing how this works, a little bit sped up. This system will load up to 15 slides at a time. It integrates seamlessly with the cell dive imager. It has 3 active processing locations to reduce the imager downtime. It can distribute reagents across the tissue dynamically with active rocking, and it can preserve antibodies and other sensitive reagents with cold storage. And you can see here the liquid handling tool within the bioassembly bot is adding mounting media to these slides on the active positions. And shortly after this, it will switch out the liquid handling tool for a plate handling tool and load slides into the imager. Maybe we'll get to see that here. And that's the plate handling tool. And it's lifting up the slide and placing it directly into the imager for imaging. So like I said, this robotic system will automate the entire cell dive process. And so it's compatible for walkaway imaging of up to 15 slides. I'll just show you some images that you can produce using this system. These are using validated antibodies produced by cell signaling technology. So we're showing 18 biomarkers here in an ovarian mucinous cystadenocarcinoma. This is a tissue microarray. We can zoom in on individual pieces of tissue here. We're looking at 14 biomarkers. Like I said before, it's a subcellular resolution. So there's a tremendous amount of detail that can be accessed using a cell dive. And so what does the user do with this tremendous amount of detail, right? Because pretty pictures are one thing, but as researchers, we need to turn those pretty pictures into actionable, meaningful data. And of course, having high quality images makes the downstream analysis easier, but we still need a means to turn those images into concrete data. And with that, I'd like to introduce Avia. It's our image analysis software, which is fully capable of working with the large 2D data sets that CellDive produces. It features automatic and AI-guided tools to cluster cells into phenotypes of interest to get a clearer understanding of the tissue. It has an easy, intuitive interface to accurately segment cells. and it offers ways to dive deeper into the biology with spatial relational tools and various charts. So using Avia with these large multiplexing data sets, you can view and interact easily with these data sets. You can detect individual cells accurately and efficiently. You can take those cells and actually use the biomarker expression data you've collected to phenotype every single cell. And then with that phenotypic data, you can generate insights and dig deep into the spatial data with interactive charts. We have some deep learning algorithms that are built into the software to make the segmentation process simpler. We use cell pose to allow for cell counting, as well as multiplex cell detection. And you can detect nuclei and cell membranes separately to get the truest picture of individual cell morphology. You can build up the phenotypes of the tissue driven by your expert knowledge by defining what phenotypes you want identified and what types of cell constitute positive cells in each phenotype. So for example, if I have expression of CD3 and CD4 past a certain threshold, I know that those are CD4 positive T cells. And obviously with these multiplex data sets and with many biomarkers being visualized, those cell phenotypic definitions can get quite fine. And it features data-driven automatic clustering to help you identify cell phenotypes that you may not know a priori. And it has a few advantages. It doesn't require you to have in-depth knowledge of all the phenotypes in the tissue. And it has the ability to easily include a large number of markers and measurements into the phenotyping engine. And it can help you identify unknown phenotypes or those you may not have considered. So this is a way to drive not only a deeper analysis of the data you've collected, but perhaps identify topics for future research as well. And once you've collected this phenotypic information, it can be a challenge sometimes to convert this into charts and present this to other users and other researchers, but Avia makes that part of the process much simpler because it has built-in data visualization tools that enable seamless insight generation and dynamic feedback on analysis results. There's linking direct back to the image, so you can click on individual cells within Avia, within these graphs, and look at the image and compare them. So with that, I'd like to turn it back to Frank for our question and answer session. And I just thank you all for your attention today. And it was great to be able to introduce these technologies.
Yeah, thanks, Michael. I will just checking the question area. So let me see. So we have on Michael or Daniel, is there a fluorescence version of the Matteo available?
I will take that question. Thank you,
Okay.
Frank. Yes, there is, which we just launched this year, which is called Matteo FL. So that is something that we added recently to our portfolio and which allows you to image 3 different fluorescent dyes or channels.
Okay, so yes, it's available and pretty new as I said, right? Okay, let's give another one. So yeah, can I use the Mica also for live cell imaging?
Yes, I will also take that one. Thank you. So Mica is both suitable for fixed samples, and I've showed you some examples of that. For example, tissues in your spatial biology workflows. But it is also an incubator, so it's actually perfectly suited for live cell imaging as well. And both because of the incubation, so to keep your cells happy over a longer time, as well as for the imaging itself, where we are able to image up to 4 different dyes really simultaneously, both in wide field and confocal imaging modes.
Okay, thanks, Daniel. So I'm just looking what's coming in. Yeah, okay, but what kind of analysis tools are available in the Mica?
Yes, so there are different types of analysis tools available, which are completely embedded in our software in easy-to-use workflows. So some of them are, let's say, more classical image analysis workflows, including intensity thresholds or different filters that you apply to your images to get to your results. Others are machine learning based tools. So we have one which is called pixel classifier, which is also completely integrated into Mika, which allows you to easily annotate your images simply by drawing in your image and thereby training a pixel classifier machine learning algorithm to identify your structures of interest. And that works really very quickly, very robustly, and with only very little annotations. And the third, let's say, set of tools are based around deep learning. So we have integrated the so-called CellPose deep learning tool, which is completely available inside the Mika software, and it allows you to do segmentation of all kinds of images that are containing cells in an automatic way. So these are the different levels of image analysis tools that are available really inside the software. And then, of course, there's also Avia, which Michael was talking more about, which can be used for any imaging data, including Mika, for example.
Okay, thanks, Daniel. Last but not least one is how many fluorophores can I measure with the mica within one sample?
So in Mika, you can't do very highly plexed samples as Michael was showing you, or as I was showing with Stellaris, for example. But with Mika, you can do 4 in one sample. So that would be in your spatial biology workflow, be more for the quality control, so to say, where you do a subset of your targets and your dyes before you then go to the highly plexed situation in CellDive, for example.
Okay, I think let me double check the one, but no more questions. But I want to come back, Daniel, on what we had foreseen as an additional poll, what we want to share with the audience in our survey, right? So maybe that's worth to lose one sentence about that. So when we send out the survey after the webinar, then it would be great to have an answer on what is currently your most challenging sample. So you have the possibility to insert a free text or a comment wherever you want to. So, and we are just curious, Daniel, myself, and Michael, to learn about what is the most challenging sample for you, because that is our job, basically, to go behind that issue and to offer you some help in your daily challenges. So it would be great if you can raise them within our survey. That brings me to my housekeeping notes and the outro. So as I shared with the most challenging sample question right now, we send out the survey together with the recording. So if you have interested colleagues within your laboratories, your working groups, your departments, don't hesitate to share that link, that all interested folks can recall that webinar. It will stay, by the way, for at least 1 year at vwr.com/events/webinars. So, and if there is any question left, don't hesitate to raise them using the webinar@avantosciences.com email address, or raise them to the experts, towards Leica and or to Avantor. We are happy to help you in doing so. So, yeah, there will be a follow-up mail, including the survey. Please don't forget us with your most challenging samples, as said before. That would be really great. And that brings me to the point to thank you all for participating in our webinar today, to thank you, our host from Leica. Well done, great insights as always. And we wish you all a great rest of the day and see you next time. Thank you.
Thank you.
Thank you.
Goodbye.